In this chapter, we're going to cover some general concepts in biochemistry, and we're going to answer some key questions, such as what is biochemistry? Why do I need to know anything about biochemistry? And then we're going to relate that to many of the things that we do in cosmetic science, and show how many different disciplines are connected together because of biology and biochemistry, and how these interdisciplinary teams work together to understand different problems that are associated with skincare and hair care, of course. Um, we'll go ahead and we'll just jump right in and try to answer these questions first. What is biochemistry? Okay, in general, when you think of biochemistry, it's a lot of people think of assays and things to measure how many proteins are in certain samples. Um, it's sort of a very specific science. You should consider to differentiate it from something like molecular biology, which is very similar, but there are key differences between them. But it's definitely different from organic chemistry, where you're doing a lot of synthetic type of work. It's different from, for example, biophysical chemistry. When we talk about biophysical chemistry, it's more like physical chemistry, so it's more of a measurement type science with spectroscopy and things, but it's focused on um, biological problems. So, for us though, what we have to keep in mind is that we're concerned with cosmetic science. So, as I told you in the introduction to um, this course is definitely geared towards cosmetic science, with specific emphasis on the principal substrates, which is skin, hair, tooth, and nail. Um, During the course of the semester, we're going to study with intricate detail the morphological and biochemical structures of these substrates. And this is really important for us, not only to know how our ingredients that are used in cosmetic products or personal care products, how they interact with the skin, the hair, the nails, or the teeth. We should also mention that cosmetic science is also concerned with the olfactory system, and this is one of our senses, our sense of smell. This is a very complex process that occurs in human anatomy and physiology, and we'll probably touch on that at some point in the semester as well. So another question you may ask is, why do I need to know anything about biochemistry? And just in general, today in science, we all need to have some type of understanding of biology and biochemistry in order to be successful in our careers. And that's mainly because, if 20 or 30 years ago, that was much different. Biology and biochemistry weren't the predominant forces in science that they are now. But everything with biotechnology, you can see all these different centers focused on biotechnology. If you look at the Boston area, the San Francisco Bay Area, the Barcelona area, right outside of London, in Germany, I think I think it's in Berlin. There's sort of a center, but these are developing all over the world because of the importance, both economically and scientifically, to understand biology. So, many years ago, biochemists were biochemists, biologists were biologists, chemists were chemists. But now, that's all starting to change, and people are starting to work in cross or interdisciplinary areas. Um, and an example of that is just to look in, any of the American Chemical Society journals, and you're very likely to find articles in the Journal of Physical Chemistry or in JACS or Journal of Organic Chemistry, where these are chemists, but they're working on biological problems. So again, I keep mentioning there are many disciplines working together in biology. We have the field of biochemistry. We also have genetics, which is definitely distinct and different. A lot of people that are biologists even say, geneticists speak a different language. We never understand them. Um, we also have, as I mentioned before, molecular biology and biophysics, chemistry, microbiology, and immunology. So, just again, to define biochemistry, it's basically the study of chemical processes in biological systems. And it deals with, essentially understanding the structure and function of lipids, proteins, nucleic acids, and carbohydrates. Okay. This list of molecules can also include other biological molecules with important functions, such as vitamins or hormones. So, another key thing with biochemistry is it essentially deals with the elucidation of metabolic pathways in the body. And, you're traditionally likely to think of things like the citric acid cycle or the Krebs cycle, or maybe oxidative phosphorylation and other different metabolic pathways. We won't really be discussing those in great detail in this class. Again, that's because this class is tailored towards cosmetic science. We're not really interested in, usually in a first semester course in biochemistry, it's very focused on all the different metabolic cycles that take place in the body. So from biochemistry, let's go to genetics. Genetics essentially is the study of genes and heredity in living organism. In addition to just looking at molecular structural studies of genes, geneticists monitor the behavior of an organism when certain genes are inserted or deleted. And you'll often hear them referred to as wild type versus mutant genes. This is looking at, when genes are inserted or deleted, that's sort of a mutant type gene. You'll also hear the expression knockout mice. This is when a gene is removed. What's probably important for us is that, in especially with relation to skin and also with respect to hair growth, mice are used for these studies. Usually for skin, a lot of the skin studies, the hairless mouse it's essentially the universally accepted model. In molecular biology, this is essentially a bridge between biochemistry and genetics, and it's essentially concerned with molecular processes in the cell. So, if you take a course in molecular biology, you're likely to really heavily study replication, transcription, and translation. So replication refers to DNA replication, so basically copying DNA, making a copy of it. Transcription refers to the transcription of a DNA molecule to an RNA molecule. This is something that we'll look into a little bit in more depth as we progress in the semester. And then, of course, we have translation, which is translating an RNA molecule to a protein structure. So this shows us the connection between DNA and DNA structure. So essentially the sequence of DNA bases that you have in a DNA chain, and how this is related to protein structure, or the sequence of amino acids in a protein chain. Biophysics, again, this is the application of physical techniques to study biological systems. And, this is especially prevalent in skincare. A lot of spectroscopic techniques, such as infrared and Raman spectroscopy, these are commonly used to study stratum corneum lipids. Another example would be using atomic force microscopy to study the detailed ultrafine structural information of the surface of hair fibers. So those are just two examples. This is the area that I have my degree in. It's in biophysics. I essentially study biophysical properties of hair and skin. Chemistry, again, just like biology, chemistry is a very broad field. And it consists of several major disciplines, as I mentioned already: organic, physical, polymer chemistry, analytical, and even nowadays we have computational chemistry. And essentially much of our understanding of biomolecular interactions and mechanisms is owed to, or we understand that based on the work that was done by previous generations of chemists. So, a lot of the modern day analytical and physical techniques that we have, these are often applied to problems of biological significance. And again, as I mentioned, the computational chemistry, this is essentially molecular modeling of proteins or any type of molecule, but with specific reference to this course, it could refer to lipids. I did a lot of molecular modeling of lipids when I worked on my degree. And essentially, the main information that molecular modeling tells you, or one of the key parts of the information, is the conformation of the molecules. And by conformation, we mean the three-dimensional structure. What does the lipid membrane look like when you insert a protein in it, or when you perturb it, or you do something else to it? So that's one of the major questions that's answered. The next discipline here, microbiology, this is closely related to immunology. It's essentially the study of microorganisms. This could include eukaryotic cells, for example, fungi, or it most often refers to prokaryotic cells, such as bacteria. And we'll discuss both of these, both eukaryotes and prokaryotes, in a few minutes. Microbiology also encompasses the study of viruses. Viruses are nucleic acids that are coated with protective proteins. And also, it could be the study of prions, which are infectious agents that contain protein that has basically an unusual tertiary structure. When we talk about the skin, why are we concerned with microbiology in the skin? First and foremost, we're concerned with the invasion by infectious agents, so pathogens, that could invade the skin and perturb the skin's barrier function. The skin has a barrier function, and essentially this could lead to some type of disease state in the skin if this barrier was compromised by these pathogens. Now, skin is actually a habitat for many bacteria. So bacteria live on the surface of the skin, and these are known as skin flora. And essentially, these protect us from foreign invasion by other microorganisms, bad bacteria, for example. So we have, these surface bacteria that are helping us and protecting us from external things in the environment. So this is similar to, we have the microflora that cover the gastrointestinal tract, inside the oral cavity region, in through the entire GI tract. It's a very similar type concept. In this case, it's on the surface of the skin. Another thing with microbiology that we're concerned about is contamination in formulations. So, a lot of times in all of the formulations that you purchase in personal care, preservatives have to be used, and that's in order to prevent bacterial growth in the finished formulations. And there's a lot of controversy about this because everyone, there's been a large movement towards natural and things, and I think most people can agree, when you can go natural, let's go natural. But when you're talking about preserving things and preventing growth from them, it's really a challenge, and it's especially a challenge when you have natural products if you don't have a preservative in it. You can imagine if you had, you couldn't, every time that you go and you use a cream, every time that you put your fingers into that cream, you're essentially contaminating it, and you really couldn't use and reuse that cream if you didn't have preservatives in it. And you couldn't keep it on a shelf. You would have to store it in a refrigerator. It would have a really short shelf life. So it wouldn't really be practical, I think, to not have something preserved. Immunology, this is a really big area, especially in relation to the skin. And we'll talk about it actually in this chapter to some extent. And, the immune system in general is a very complex system that protects the body. It helps the body identify and protect itself. At the same time, it kills foreign agents and tumors. The skin in particular is an outpost of the immune system. And immune cells go in and out and travel through the skin. There's a lot of molecular signaling that takes place when we have attacks by foreign species or foreign pathogens. It's really an incredible outpost of the immune system, and capable to launch an actual immune response in the event of attack by a foreign pathogen. So let's just briefly talk about some important events in biological fields and what impact these have had on cosmetic science. So the first one here is sequencing the human genome. So you might ask yourself, what does that have to do with cosmetic science? Just in general, genome refers to all of the genes or genetic material in a cell or organism. And I don't know if, depending on your age, I'm not sure if you'll remember this, but back in the early 2000s, this was like a really big thing, was, all the talk about sequencing the genome. It was something new, exciting, and I think by understanding the sequence of the human genome, we thought, this would help us understand disease better and how the body functions better, and I think it has to a great extent. Again, we mentioned gene. When we talk about genes, a gene is essentially a series of 3 nucleotides. A nucleotide is essentially the monomeric unit that constitutes the backbone chain of a DNA molecule. And various combinations of 3 nucleotide units correspond to an amino acid. Okay? And just like we spoke about before, when transcription and translation occur, this ultimately leads to protein synthesis, and the order of the genes along the DNA chain will produce a protein chain with a corresponding amino acid sequence. So this is how the DNA sequence, the gene, each gene, which consists of the 3 nucleotides, the sequence for 3 nucleotides is a code for each amino acid that's in the protein. Okay? And as you go along the DNA chain, the first 3 nucleotides correspond to one amino acid, the next 3 after that to another amino acid, the 3 after that another amino acid. So you can see for 9 nucleotides, you only have 3 amino acids, a sequence of 3 amino acids. But this would continue on as you go along the DNA chain. So, with regard to the skin, why is sequencing of the human genome important? One of the key things, or one of the key studies that was completed not too long ago, it's been almost 8 for the fungus Malassezia globosa. That's in it. Malassezia globosa. If you need the spelling of that, you can check the handout that I'm going to send you with this video. This is essentially the fungus that's responsible for the formation of dandruff. Okay, you may ask, why is this fungus so important? It's known to play an important role in both psoriasis and eczema, 2 skin conditions, as well as atopic dermatitis. And, the idea is that, in general, understanding or having some knowledge of the genetic composition of this fungus could help scientists understand how this fungus interacts with the skin, and it may help with the development of future treatments. The next thing on this list, or the next important event, great strides in immunology. This is what I mentioned before, that I would just like to sort of talk a little bit about the connection between immunology and skin. And, if you think back, and if you look back into the literature prior to the 1970s, essentially skin wasn't really considered that important of an organ. Now it's considered an important organ. It's actually the largest organ in the body. And, it wasn't until maybe the early to mid 70s, and actually into the late 70s, that people started to realize that there was some connection of the skin with the immune system. And essentially nowadays, why this is so important is because we have a field known as photoimmunology. And photoimmunology is essentially understanding the interaction of light with the skin and how this affects the immune system. I just like to briefly mention, there was Professor Margaret Kripke, Kripke, and you should look in your handout for the spelling and just a little review that's written about this. But essentially, she's credited with understanding a lot of what we know between the relationship between the immune system of the skin and skin cancer. And I'm not sure if I have the figure here. I don't believe I do. But essentially, she did experiments of mice where she subjected the mice to UVB radiation, causing them to develop tumors. Okay. Now, she took the tumors from these mice and she transplanted them into 2 different sets of mice. One set was non-immune suppressed mice, so just normal mice in the laboratory. that were not immune suppressed, so they weren't exposed to UVB radiation or any other agent that could cause immunosuppression. And then the mice, and then tumors were transplanted to mice that were immune suppressed. And there were actually two groups in the immune suppressed mice. One group was subjected to sub-carcinogenic doses of UV light. So, UV light, but it wasn't strong enough to initiate a carcinoma event, so cancer. And then the other group was actually immune suppressed with immunosuppressive drugs. So essentially, there are certain chemicals or drugs that you can give to the mice. You can suppress the immune system. And, their findings were revolutionary in as much that they found that the tumors did not survive in normal mice. So the non-immune suppressed mice, the tumors could not survive. So that means that in those mice, when you took tumors and put them in their skin, transplanted them in their skin, essentially their immune systems were in good shape, and they fought off and killed the tumors. But what they did see is that in the immune suppressed mice, both the UV exposed mice and the mice that were treated or given the immunosuppressive drugs, they found that the tumors were basically highly antigenic. So they stimulated the production of antibodies, and the mice in those groups were unable to kill the tumors off. The tumors were able to survive. So this, the importance of this is that it provided a demonstrable link between photocarcinogenesis and immunity. And, this is what led to the birth of a new field in photobiology, which is today known as photoimmunology. And essentially, this is a very important field. If you look in the literature, there are many journals actually dedicated to this area, and there's just a lot of research going on worldwide in this area to better understand this still. A third event is the damage to the ozone layer. The ozone layer, as you know, there's ozone that's in the Earth's atmosphere. So ozone is O3, the actual chemical formula. It's present in the stratosphere, and it's important there because that's what filters out a lot of UVB light. As you know, UVC is pretty much entirely filtered out. UVC light would be extremely dangerous. Thank God that it doesn't reach the surface of the Earth. UVB, though, on the other hand, there's a fair amount of UVB light that reaches the Earth, but a lot of it is filtered out by the ozone layer in the stratosphere, so in the Earth's atmosphere. Another thing is, if that's damaged, UVB light can get through to the surface of the Earth, and it can reach us and essentially damage our skin. So it can damage DNA, it can damage lipids, and it can also damage proteins, not only in the skin, but also in the eyes. And essentially the danger here is that this UVA and UVB light that reaches us, the surface of the Earth, can be carcinogenic. Just a note on this, we're talking about ozone in the ozone layer. We should also note, though, that while ozone in the ozone layer is important to help protect us from incoming UV light, ozone that's at the surface of the Earth is actually bad for us because this is ozone that's caused by pollution. This essentially causes free radical reactions And of course, you can imagine this has damaging effects on the skin. There's also major concerns because it has um, inhalation issues. So you can imagine if you have these reactions taking place in the lungs and other tissues of the body as well. Another important event is the destruction of the rainforest. Um, there's a lot of controversy about this, especially in large countries such as Brazil and Indonesia that have very large percentages of rainforest. Essentially these rainforests are extremely important for the production of oxygen on the earth. But in addition to this, the ecological systems that are present in the rainforest are just phenomenal. Um, the different life forms that are present in the rainforest is just it's just amazing. If you've ever watched documentaries on the rainforest, it's just amazing. All the different species of of animals and insects and other beings that are present in the rainforest, and not to mention all of the plant life that exists there as well. So a lot of times though these are developing countries, so the economics are such that these countries are looking for ways to earn more money. So a lot of times they're logging or they're conducting agriculture or mining in these rainforests and using them for industrial use. Um, and this is really leading to the destruction of the rainforest, and this is killing off species. And again, the importance of having all of these species is that we have a great deal of biodiversity. Another thing to mention is there's a lot of diverse vegetation in the rainforest, and again, this is really important. If you look, a lot of the plant medicines come from rainforest, and as I mentioned already, a large percentage of the Earth's oxygen is synthesized by plant life in the forest. One of the major crops that's occurring in the rainforest is palm oil, and that's, I sort of mentioned that because palm oil is something that's used extensively in personal care or in personal care products. So that's something just for us to think about in the future. Where is this palm oil coming from? Is it coming from the rainforest? Are there alternative ways that we can go about using this or harvesting it, or even are there alternative molecules that can be used instead? The last point on this slide is cellular signaling. And essentially, this is a field that has exploded in recent years. It's a discipline in biology, and essentially, just in its most basic form, it refers to a science that discusses the secretion of a molecule by one cell into extracellular space, and then sending that molecule to another cell where it binds to a receptor on the cell membrane. And by binding on the receptor, it leads to a chain of events, molecular conformational changes in different molecules in the cell. But the ultimate is that it leads to the transcription and the eventual expression of a molecule. So, one cell secretes this molecule. The molecule goes from that cell surface to another cell surface. It binds with the receptor. There's a chain of events that happen in the cell, but essentially you have signaling that occurs, and the message is sent to the nucleus of the second cell, and this tells it, okay, we need this protein synthesized or upregulated. So it leads to to transcription first, and then to translation to a new protein, and then some event that cell is going to do. So what this allows cells to do a lot of times is they can act in unison. So they can sort of act together as a chorus, for example, and they can do many different things. And I'm going to review a couple of things here with you. We'll go ahead and we're going to talk about the cell next. Okay, so now let's talk a little bit about the cell, and we're essentially going to talk about three distinct life forms or domains of life that are on Earth. The first one is bacteria, and bacteria, you typically think of soils, surface waters, and tissues of organisms. Those are the places where they are found. Typically, bacteria are single-celled organisms, and they're classified as being aerobic or anaerobic. So aerobic would refer to bacteria that metabolize oxygen, or they live with oxygen, and anaerobic would be those that live without oxygen. Another distinct domain of life is archaea, and these organisms exist in very extreme areas, extreme meaning extreme environments, such as salt lakes, so places where the salt concentration is really high. Just to give you an idea, when you're talking about the ocean, just roughly in general, you're, I think, referring to probably about 3% in terms of levels of salinity. If you look at like the Dead Sea, which is one of the highest places in terms of its degree of salinity, I think it's close to like 30%. You're actually really, if you go in the Dead Sea and you swim, you'll notice that you're very buoyant. You have to be really careful not to get the water in your eyes because of the very high levels of salt. But archaea can also live in hot springs or in the very depths of the ocean. Very high pressure, very cold regions. We also are going to talk about eukaryotes, and eukaryotes can refer to several different types of cells. We'll talk about animal cells, and that's going to be a focus for the semester that we're going to be concerned with. We'll also briefly mention the difference between animal cells and plant cells, and then, just to mention another group, fungi are also eukaryotic cells. So I thought I would just mention Dr. Bassler. She actually studies what's known as quorum sensing, and I just wanted to mention this because she actually studies how microorganisms send molecular signals to each other, how they conduct this essential quorum sensing, and how they can sort of launch an attack, or how they can operate in unison with each other. So this is just more of a general information. You don't really have to know anything about this, but just to sort of let you know, like, people are actually focused on this type of research area. So let's jump right in and look at the bacterial cell first. So the bacterial cell is known as a prokaryote. So bacteria are, in general, prokaryote cells. And here we have a list of the different components that you should be familiar with. So, if we start on the outside of the cell, We have what's known as a capsule here. Then we have a cell wall, and then a plasma membrane. So if we look at the three of these together, this is actually known as the cell envelope. Okay, so the capsule, the cell wall, and the plasma membrane are known as the cell envelope. And together, these three components provide a barrier for the interior of the cell to the outside world. Okay, so the plasma membrane, this is essentially analogous to what's found in animal cells or in eukaryotes. We'll talk a little bit about that. It contains lipids that are embedded with protein structures. And again, the lipids form like the lipid bilayer of the cell membrane. But up until recent years, people kind of forgot to mention that, there's a lot of protein structures that are embedded into that. And these protein structures essentially serve as channels for things to go from the outside to the inside of the cell, or vice versa. And also receptors, as I mentioned before, where you can have receptors where other external molecules can bind to the outside of the cell and sort of cause some type of reaction or occurrence of an event to happen within the cell. Okay, just in general, the cell wall helps the cell maintain its rigidity. So it's a strong cell wall, and it helps it maintain its shape as well. And this is comprised of peptidoglycan, that's spelled PEPTIDOGLYCAN. So it's peptidoglycan, and this is essentially a biopolymer that contains peptides and carbohydrates. We'll mention something about that a little bit more on one of the following slides. But essentially, I just wanted to define these parts of the cell, and then if we look also on the outside of the cell, we have what are known as we have the bacterial flagellum. Essentially, this is this little tail here. This helps the bacteria move around. As you can imagine, it's sort of like swimming around. You also have these little sort of things that are projecting out from the surface of the cell. These are known as pili, P-I-L-I. This is what allows the cell to attach to a surface. So even if it's a tissue surface, this helps it anchor in and attach to the tissue there. On the interior of the cell, we have several important components. First and foremost here, we have the nucleoid. This is essentially a circular type of DNA. I'm sorry, usually this is a double-stranded string of DNA that's in the circular form. Okay. So it's called a nucleoid, but just to keep, the difference between eukaryotic cells, we'll see in those we actually have a nucleus that contains a nucleolus in the eukaryotic cells. So we have in bacterial cells we have nucleoids. We also have this blue, all the blue that you see in here, all the background. This is known as the cytoplasm. And essentially, this is like the aqueous part inside of the cell. So you can imagine it's, this is like a solution for the most part. Cytoplasm is what it's usually called. It's also, you may also hear it referred to as the cytosol. The cytosol actually refers to the aqueous solution itself. So the cytoplasm is like all of the interior of the cell with the organelles and everything. The cytosol is the aqueous solution. We also see that there's ribosomes here. The ribosomes are important for protein synthesis. This is essentially where protein synthesis takes place. And, just to note, the ribosomes of bacterial cells are different than ribosomes from eukaryotic cells or eukaryotes. They're smaller in prokaryotes. Okay. And, another thing that's shown here, as you can see, are plasmids. And plasmids are another distinct form of DNA that are present inside of the bacterial cell. So, we mentioned the bacterial cell wall and how it contains peptidoglycan, which is shown here, the spelling of it. And essentially, this is a biopolymer that consists of both peptides and carbohydrates. And essentially, if you have bacteria with thicker cell walls, they're going to have greater quantities of peptidoglycan. So, you'll hear of bacteria that are called gram-positive or gram-negative bacteria. Essentially, gram-positive and gram-negative refers to how the bacteria react with a stain. It's a biological stain. It's actually a purple stain. It's called Graham's stain. And, you can see in this image that's shown here on this slide, the gram-positive bacteria are these, more purple ones that essentially interact with the dye. And they interact with the dye because they have greater quantities of peptidoglycan, and that interacts with the Gram stain. The gram-negative bacteria don't interact as much, and you can see they don't have the dark purple color that the gram-positive bacteria have. And just in general, there's a lot more that we could discuss about this, but it's differences between the cell wall structures that differentiate these two types of bacteria. Just to keep you sort of aware that these exist. You could go much more in depth into them, into the structure and function of these, but we won't for the purposes of this class. So now that brings us to animal cell structure. So now we're going to talk about eukaryotes. And here you can see that there are a number of different important structures present here. So we'll sort of start with in the middle here, I think, and we'll talk about the nucleus first. So the nucleus is this sort of pink region that's shown here. Then we have the nucleolus, which is the center of the nucleus, and then you even have the nuclear envelope, which is essentially it's like a membrane for the nucleus. Okay, so we have those three things going on there. The nucleolus is essentially where ribosomal RNA synthesis takes place. Okay, so remember, when we talk about the nucleus, DNA replication, which is, the copying of one DNA and making a second copy of it occurs. It's also, the nucleus is also the site of RNA synthesis, which is known as transcription. Okay, and then when we talk about translation or protein synthesis, this actually occurs in ribosomes. And you can see here the ribosomes are these little circles or spherical type shapes that are shown here, and they're mounted on the rough endoplasmic reticulum. And it's called rough endoplasmic reticulum because it has these ribosomes that are studded over it. Okay. Once proteins are synthesized by the ribosomes, they're then transferred from the ribosomes to the Golgi apparatus. Okay, and I don't see the Golgi apparatus noted on this, but you should know that. Golgi is spelled Golg. Ah, it's here. I'm sorry. Golgi complex is shown here. So they go from the ribosomes. The proteins are sent to the Golgi apparatus, and they're essentially in vesicles. So they're kind of like in little organelle type vesicles. Okay. Then from the Golgi apparatus, the proteins are transported to their final destinations. We also have the smooth endoplasmic reticulum, which is shown here adjacent to the rough endoplasmic reticulum. This is where lipid metabolism and drug metabolism takes place. We also have peroxisomes. You can see this there's one shown here. There's actually several others that are shown within the cell structure. And these essentially contain enzymes that are responsible for getting rid of toxic peroxides that are in the cell. Also important that there is fatty acid oxidation that takes place in the peroxisomes, also known as beta oxidation. But we should keep in mind that beta oxidation, that's not the major place where it takes place. It actually takes place, the major place that it takes place is in the mitochondrion. And we should also mention that there are lysosomes that are present in the cell, and these contain digestive enzymes that break down food particles, also old organelles, viruses, and even bacteria. So, you can see that you have these things like peroxisome, lysosomes. They end with SOME. These are responsible for breaking things down and getting rid of things that the cell doesn't want. Now the mitochondrion, which on here, let's see if you can see, it looks like a little sausage here. This is how it's drawn when it's illustrated in textbooks. If you look at it in real life, it actually wraps all around the cell nucleus here. It's all over the place within the cell. The mitochondrion is essentially the energy factory of the cell, and it's responsible for the formation or synthesis of adenosine triphosphate, also known as ATP. ATP it's the essential energy source for the body. Whenever any process or any biochemical process takes place that requires energy to synthesize certain molecules, ATP is like a primary source of that. So we'll briefly discuss that or go over that in a couple slides. So I think just in general we should talk about the mitochondrion as the energy factory of the cell. So the synthesis of ATP, this is known as oxidative phosphorylation. It's a process that takes place, and we'll probably discuss it a little bit more in detail later in the semester, but we'll give you a brief view of it. But some other important metabolic processes that take place in the mitochondrion are beta oxidation, okay, which we mentioned. This is fatty acid oxidation. Also the citric acid cycle. This is a metabolic cycle that's also known as the Krebs cycle or the tricarboxylic acid cycle. And then there are also amino acid oxidation pathways that occur in the mitochondrion. So for ATP synthesis, this is ATP or adenosine triphosphate. Adenosine, as you will see as we progress in the semester, this refers to this base that's shown here. This is also present in DNA, this particular base. This is the base structure of ATP, and then you can see there's this ribose structure that's shown here, and then you have the triphosphate unit that's shown here. So As I mentioned before, a lot of times energy is produced when this undergoes a reaction and this phosphorus oxygen bond is cleaved here. And that's what leaves cleavage of that bond releases a fair amount of energy, and it helps synthesize or catalyze different processes in biochemistry. So we also mentioned beta oxidation. You know, this is essentially an important metabolic process, and part of it encompasses the conversion of fatty acids to acetyl-CoA. Just to give you a reference, acetyl-CoA, you should be familiar with the ATP structure that I showed you. You don't really need to worry about acetyl-CoA right now. This isn't as important, but it's just to illustrate to you, the complexity can continue to arise in biochemistry. Here you can see there's this another base type structure, ribose structure. Then you have two phosphate groups, and then you have this, whole chain of molecule here that's end capped with this sort of acetyl group. This bond here that contains the sulfur, if you'll recall, an ester bond is when you have an oxygen here, and then you have O, C, double bond O, and then you have R and R on the ends. This is actually a thioester, so when you have an S here instead of an O. And the reason I point that out is that we're going to talk about an important thioester bond that occurs in the hair fiber at some point in this semester, during this semester. So I won't really say too much more about these last two cycles. The citric acid cycle, we're not really going to discuss that for the purposes of this class, nor the amino acid oxidation pathways. If you can check out a general biochemistry book from the library, and you can sort of get an idea of what these pathways are concerned with. But essentially, it just goes through changes that occurred during the two cycles. So another thing to mention about eukaryotic cells are the plasma membrane, or is the plasma membrane. And essentially the plasma membrane separates the interior part of the cell with the exterior world, or the exterior environment. It plays a really important role for the passage of ions, also water, and even other molecules that go from the exterior of part of the cell to the interior of the cell, or vice versa. Okay, as I mentioned already, it's primarily composed of phospholipids, but you can see here in this illustration the phospholipids. This is the phospholipid head group that's shown here, and then you can see the tail that's shown here. So a bilayer is formed when you have this top layer. So you have like one top layer of phospholipids that interacts with the tails of a second bottom layer of phospholipids. That forms a bilayer type structure. But you can see, in addition to the bilayer or the phospholipids that are present in the bilayer, you also have other types of proteins. You can see here's like an alpha helical type protein. Here you can see this other type of protein structure. This you can see is an integral protein. This has a globular type protein structure. Globular just means when you talk about protein structure, it sort of has a globular overall three-dimensional structure. You can see that some of the proteins also they're not just proteins, but they're glycoproteins, and glycoproteins contain both carbohydrate and protein molecule together. You can see that Here you have the carbohydrate portion coming off of this molecule. Um, Cholesterol is also an important component of the cell membrane. You can see that it's shown here. And the reason it's so important is that it adjusts the fluidity of the membrane. So it can either make it, when you have a cell membrane, you don't want it to be too rigid or stiff, but you don't want it to be too soft either. And cholesterol plays an important role with adjusting this to the right amount or the right extent. Also shown in this image, you can see this is the interior part of the cell down in here. Up here in the top, this would be like the exterior part of the cell. So in the interior part of the cell, we have what are known as the cytoskeleton. So the cytoskeletons, or the cytoskeleton of the cell, essentially consists of three types of molecules. One is actin filaments, ACTIN, and then the word filaments. The second one, microtubules, that's MICROTUBLES. And then the third one, or the third class, is intermediate filaments. Intermediate filaments. And intermediate filaments, these actually refer to keratin proteins, and we'll be talking a lot about these throughout the semester. But just in general, in a regular cell, a regular eukaryotic cell, these filaments essentially are what form the cytoskeleton of the cell. They give the cell the structure that it has. They help it maintain, certain level of rigidity or softness, whatever it is, in that particular cell. And they are also responsible, for helping the cell modify itself as necessary when it has to undergo very important processes such as mitosis. So you can, if the cell has to change shape, these can help it change shape. You can have certain levels of, you can adjust the levels of these filaments would be adjusted to a certain degree to make a cell more flexible, for example, when it undergoes mitosis and you have cell division. And we'll talk about these a lot with regard to the skin, and you'll see how, as you go from the very bottom layers of the skin to the very top layers of the skin, that the filaments that essentially form the cytoskeleton of the cell changes dramatically as you start at the very lowest level of the skin. This is known, or the epidermis actually, until you get to the very outer layer of the epidermis. We'll have that for another day, but for now, let's just shift gears a little bit and talk about plant cell structure. So again, a plant cell is also a eukaryotic cell, and one of the things that we should mention that's really notable in a plant cell is its cell wall structure. It has a very strong cell wall, much stronger than you find in animal cells, and this helps protect the plant cell from what's known as osmotic swelling. Osmotic swelling, osmosis in general refers to the flow of water through a semi-permeable membrane, basically from one compartment to another. So if you have, usually you refer to one compartment to the other. If one compartment contains a higher concentration of salt or solute, you would have flow from the compartment that had lower levels of solute to the compartment that had higher levels of solute. But what could happen is that, the passage of water, for example, could go into this other compartment and start swelling this compartment to an extent that it would essentially, or could eventually, undergo cell lysis, which is basically bursting of the cell because you've overloaded it with the quantity of water. In addition to that, we have plasmodesma that are present in this type of cell wall structure. You can see this is shown here, and this essentially serves as a pathway between cells so that you can have nutrients going from one cell to another. Plant cells also contain vacuoles that you can see this here that's shown, and these are essentially storage compartments for food, waste, and even water. They can also degrade and recycle macromolecules. So that's a pretty important function in the plant cells. We also have glyoxysomes that are present in these cells. Essentially, glyoxysomes are organelles that contain enzymes of what's known as the glyoxylate cycle, and this is a metabolic pathway that produces oxaloacetate that's needed for gluconeogenesis, which is carbohydrate formation. So this is analogous to the mitochondria in animal cells. You can see here that it's shown, a mitochondrion is shown in this. What's also given here are chloroplasts. Chloroplasts essentially are the energy source of the cell. Again, in the case of plant cells, chloroplasts are sort of more at the forefront, kind of like mitochondria are in the animal cells. Chloroplasts are in the plant cells. So they're the really important part, and this is where the cell, where we have the formation of ATP and carbohydrates in the cell, and this is where photosynthesis takes place, where we basically convert solar radiation energy. There's absorption by a class of molecules known as cytochromes, and this is converted into biochemical energy through the synthesis of ATP here. So, just to even get into a little bit more depth, you can see these little stacks of they look like stacks of coins that are shown in here. This is actually where photosynthesis takes place inside of the chloroplast. You basically have these membrane-bound organelles that are known as thylakoids, so it's spelled THYLAKOIDS. And then, the last thing that we should mention here, I'm not sure if I see it here so much, but starch granules are present in these types of cells. And the starch granules are actually storage areas for carbohydrates that are formed as a result of photosynthesis. Okay, so just to go back, I know some of the terms that I mentioned you don't see on the chart. Starch granules isn't shown here. Thylakoids. So thylakoids again is spelled THYLAKOIDS. Chloroplast is shown on the chart. Glyoxysomes are not shown, or that term is not shown on the chart. The vacuole is shown here. Plasmodesma is not shown on the chart. Cell wall is cell wall is shown on the chart, I believe. At least you can see the cell wall is shown here. Essentially all of this that you can see. Okay, and then you also see that this type of cell has a nucleus that consists of a nuclear envelope, nucleolus. The chromatin chromatin and nuclear pore. I mean, this is similar and analogous to what you see in the animal cells. So let's talk a little bit about water and pH. And the reason why is water is very important in biochemistry, and just in general, life on Earth, water is like 70% of the Earth's composition, and that's just in the form of oceans, seas, glaciers, lakes, and rivers. But even with humans, I mean, the body contains about 70% water as well. So essentially, water is the universal solvent for all the proteins, all the biomolecular machinery that keeps us functioning as an organism. Okay? And the three-dimensional structure of not just proteins, but also lipids, carbohydrates, and even nucleic acids all depends on the presence of water. So we'll talk about several different things in relation to water. First, we'll talk about hydrogen bonding. And you can see there's a list of other things looking at water as a solvent. We'll talk about extracting lipids from hair. I know this is sort of out of place in this list of things that we're discussing, but it's something that I wanted to cover in chapter 1, and then it'll sort of give us some understanding of things in later chapters. We'll go over some definitions, talk about equilibrium and pH, and then why is pH important in cosmetics? And then we'll talk about pH, acids, and bases, and then we'll also talk about buffers. And you'll see the reason why we want to talk about buffers is not just from an experimental standpoint, just to, whenever you conduct a lot of different biochemistry experiments, you need to understand about buffers, but also, all of the proteins that are present in the body, they act as buffers, and we'll see that throughout the semester. So looking at hydrogen bonding first, hydrogen bonding, I remember when I first learned about it, I thought, oh, that's nice, but what does that really have to do with anything? You're going to see in this semester that hydrogen bonding is extremely important. Just in general, hydrogen bonding, you have different types of what are known as non-covalent bonding that take place in everything. Okay, when we talk about non-covalent bonding, you have hydrogen bonding, you have van der Waals interactions, and you also have electrostatic interactions. So electrostatic interactions are when you have a positive charge and a negative charge. Some people call these salt bridges. Van der Waals interactions are when you have, you can have different molecules that are just sort of attracted to each other at some point, and depending on the distance they are from each other. And sort of related to this is hydrophobic bonding, where you have hydrophobic groups that are sort of attracted to each other, and they exclude water. That's sort of a form of van der Waals type of interaction. But the hydrogen bonding, just to give you an idea of it, it's really important in the structure of hair, for example. And if you notice, like if you try to cut hair or pull on it, like if you try to extend it when it's dry, it's much harder to do in the dry state than it is in the wet state. And the reason for that is the hydrogen bonding that takes place inside the structure of the hair. When you wet the hair, essentially the water molecules disrupt all of the hydrogen bonding that's present in what's known as the matrix part of the hair. And when they break those bonds, what happens is that you essentially have a weaker overall structure, and that's why if you try to cut wet hair, it's much easier than dry hair, and it's also why if you try to extend or like pull a hair fiber apart, it's much more extensible in the wet state than it is in the dry state. So just in practicality, if you think if you comb or brush dry hair and you arrive to a knot, you'll kind of stop where the knot is. If it's wet hair, it'll let you pull a little bit further with it. So that's showing you that it's more extensible in the wet state. Just for the purposes that we're showing you here, this is just showing you hydrogen bonding that occurs in water on this slide. Essentially, you can see the water molecule contains an oxygen and 2 hydrogen atoms, and neighboring water molecules hydrogen bond together, and you can see that the oxygen from this interacts with the hydrogen atom from another molecule, and then this oxygen this oxygen on this molecule here interacts with the hydrogen from another one. And you can see, all throughout here, hydrogens and oxygens are interacting together in a form of hydrogen bonding. In biological systems, a lot of, at least in biochemistry, a lot of the things that we'll talk about during the semester, will essentially, you'll have these hydroxyl groups, we'll also have, sort of carboxylate type groups. You'll have nitrogen containing groups, oxygen containing groups. Essentially, you can see here where these interact with a hydrogen bond donor, so you have like the OH group here. This interacts with oxygen. It also interacts with nitrogen, so you can have hydrogen and nitrogen interacting together. In this case, the top ones here are the hydrogen bond acceptors. The bottom ones are the hydrogen bond donors. Again, another type of situation that we can have, if we have similar hydrogen bond acceptors that are shown here. Actually, these two are reversed in this second illustration below, but you can see we have an NH group, and you can get hydrogen bonding between this hydrogen that's attached to nitrogen with the oxygen in both cases here, and also with the nitrogen that's shown here. Okay, so just to get a little bit more into depth with that, if we have an example here that's shown with a water molecule, and then we have oxygen. This can interact with an alcohol group. So if you have an amino acid that has a terminal OH group, which there are a couple of those, they can interact with water, as you can see here. Okay. You can have a ketone group shown up here at the top. This can also interact with water. And again, these are just examples, just a few examples to give you a flavor for this. But you also have, like, between two peptides or two proteins, you could have, a ketone type group here. So you have CO. This oxygen can interact with the hydrogen that is attached to this nitrogen. So this would provide sort of an interaction between these two protein chains that would provide some type of adhesion or cohesion between these two molecules. Here you can see we have the two DNA bases, thymine and adenine. This is just showing you the base structure. Before I showed you there's like a base structure, a ribose structure, and then a phosphate structure. The R group would be the ribose and the phosphate structure in both cases. But then what actually interacts in the DNA that gives you the alpha helix that you are so familiar with is the interaction. What stabilizes that alpha helix is these hydrogen bonds that occur between the bases. And you can see, you have an NH group here, and this is hydrogen bonding with this nitrogen that's on the internal part of the ring. And then you have another hydrogen bond between the ketone oxygen here and the NH, one of the hydrogens on the NH2 group. So this stabilizes, basically complementary DNA bases. So then that brings us to H2O as a solvent. Okay, and just to briefly, we already mentioned this, I think. It's a polar solvent that dissolves most biological molecules. If you have a hydrophilic molecule, okay, that'll be like your solute. It'll easily dissolve in H2O. If you have a hydrophobic molecule, it is not going to dissolve easily in water. So, just in contrast to that, if you have a nonpolar solvent, it will not dissolve a hydrophilic molecule, but it will dissolve a hydrophobic molecule. So just, water is a hydrophilic molecule, it's a hydrophilic solvent, so it's going to dissolve hydrophilic molecules. It's not going to dissolve hydrophobic molecules. But if you have like a nonpolar solvent would be like hexane or something like that. Hexane will dissolve a hydrophobic molecule, but it will not dissolve a hydrophilic molecule. Okay. So let's go to talk about extracting lipids from hair. So, just to give you an idea of how to do this, first, hair has a lot of lipids present in its structure. It has lipids that make up or constitute its intricate, ultrafine structure. So there's certain types of like lipid membranes present in the hair structure, and they basically keep things from getting into the hair and getting out of the hair, similar to what occurs in the skin. But they also present in hair are sebum lipids, and these can be found in the internal part of the hair and also on the external surface of the hair. And sebum lipids are really important. They offer a protective mechanism for the hair. Having them on the surface provides a very nice, rich, hydrophobic surface that keeps a lot of things from entering and damaging the hair. So, a lot of times we're interested in studying, what happens to the hair when you remove the lipids, because a lot of times if you look at like processes that the hair undergoes, such as bleaching or dyeing, permanent waving, even relaxing the hair, all of these processes damage the lipids in the hair, and they drastically change the surface energy properties of the hair, such as, if you look at like the surface tension of the hair, it's or the contact angle, it's much lower for hair that has been chemically treated than hair that's virgin hair or hair that hasn't undergone any chemical treatments. And the same is true with hair that's been, where you remove the lipids, it has properties that are similar to hair that has undergone these damaging treatments. So in this slide, what we show here is a soxolite extraction unit, and essentially, this doesn't contain hair. This is just showing you the example. But usually, you would put the hair in between. You can see there's like these. I don't know if it's cotton or nylon swabs here. We actually have like a little thimble that we usually put the hair in, and then we just place it in here. And then what happens is you put your solvent down here, and you heat the solvent evaporates, it basically goes up this tube and then goes up here, and then when it reaches up here, so it's in the vapor state, and when it reaches up here, you have a condenser up here, so it goes up into this headspace, but then it can't go any higher because it has to condense because of the cool temperature here. So it condenses, falls onto the hair, which is in here, and then it extracts lipid components from the hair and then carries the lipids down into the main solution down in here. And so each time this process occurs over a period of cycles. Each time that you have solvent that's evaporated up into here, this solvent is free of lipids, so it's kind of like you have fresh solvent that goes and pours down over this, and then it keeps all of the lipids down in the bottom section of the round bottom flask that you see here. Okay, this gives you some other information, and you have this in your handout as well if you want to refer to this again. But that's essentially how the technique works. Usually what you do for a oxalate extraction procedure is, at least for most tissues and particularly for hair, you usually start with a non-polar solvent first. So hexane is a non-polar solvent. You would extract the hair first with this. Then you would change the solvent. You would use a slightly more polar solvent. This would let you, the idea is that the non-polar solvent would remove everything from the surface of the hair. If you use a solvent that has a little bit more, or it is a little bit more polar, it'll start to remove some things from the internal part of the hair, because remember that the hexane really doesn't penetrate the hair. Tertiary butanol will a little bit, and then if you use a mixture of chloroform and methanol, this is a more polar solvent. This enters into the hair, and this will remove a lot of the lipids from the internal part of the hair. Okay. So we'll kind of talk about like lipids and things with hair and why that's important in a later chapter. But I just wanted to sort of give you an introduction to the oxalate extraction technique in this first chapter. Now let's just go over a few definitions with regard to water and pH. So we have amphipathic compounds. These are essentially molecules that contain regions that are polar or charged, and also nonpolar. So it has both polar and nonpolar areas in the same molecule. That would be an amphipathic molecule or amphipathic compound. Hydrophobic interactions, as we mentioned, these are forces that keep nonpolar regions of molecules together. Okay, so if you have like a long aliphatic chain on one molecule and then you have one on another, those two chains or two alkyl chains or alkyl groups would sort of interact together and stay together. We should also mention a few things about osmosis. So, first, osmosis, as I mentioned before, it's the passage of water across a semi-permeable membrane, which is driven by differences in osmotic pressure. Okay. Osmolarity is a measure of solute concentration. Isotonic is when the osmolarity of the solution is equal to that of the cell. Hypertonic is when the osmolarity of the solution is higher than that of the cell. And then hypotonic is when the osmolarity of the solution is lower than that of the cell. So, just in general, cells contain high concentrations of biological molecules and ions, and they have to sort of address and take action to look at the differences in osmolarity between the cytosol of the cell, and the surrounding solution. If the cell didn't do anything, H2O could enter the cell eventually until it swells to a point that it would burst, and then it would undergo a process known as lysis. So you really wouldn't want that, because then you would have damaged cells. If you look at the plant cells, and we kind of talked about this a little bit, or bacterial cells, they have a very rigid membrane, and that prevents lysis from occurring. But if you look at animal cells, another sort of thing that we have to look at or remember here is in animal cells, or in animals in general, we have extracellular fluid, such as blood, and we also have interstitial fluid, and these are maintained at constant osmolarity. So this helps us from basically having cells that undergo lysis. In addition to that, though, we also have, our cells are basically equipped with ion pumps. So I mentioned before, you have ion pumps in the cell membrane or the plasma membrane. And these basically transfer ions from inside to outside the cell, or vice versa. And the transfer of these ions essentially ensures that the osmolarity remains the same on the inside and the outside of the cell. Okay, but just, in general, in biochemistry and molecular biology, you're often working with cell cultures. So whenever you do those experiments, you usually carry out the procedure in an isotonic medium. Okay, so that brings us to equilibrium and pH. So just, we should mention that when we talk about water, it doesn't actually fully exist as the molecular entity that we envision, but instead, it has the tendency to ionize into its component ions, and you can see that on this next slide here, where in equation 1 here we show, H2O actually dissociates into H plus OH minus. In fact, that's what we usually write down in all the equations, but there's actually the H plus ion exists as what's known as the hydronium ion, and that's an H3O plus. Okay, so why are we even discussing that? Well, we want to talk about pH a little bit, and essentially when the pH, or when the concentration of hydrogen ions and hydroxide ions are equal, the solution will be a neutral pH, so pH 7. If you have high concentrations of hydrogen ions, or H+, the solution will be more acidic, and it's going to have a lower pH. Well, if you have lower quantities of H+, you're going to have more basic solutions, and the pH will be greater than 7. So if you look at these equations, equation 2 that's shown on this slide essentially shows you how the pH of a solution can easily be determined if the concentration of H plus is known. Something else we should note is that the pH scale is logarithmic, so if you have a difference of 1 or a few units, that difference is much greater than what you might think it is. Just in general, some acidic substances include hydrochloric acid that has a pH of 0, vinegar pH 2, cola, such as Coca-Cola, has a pH 3, beer has a pH 4, but in contrast to that, you have basic substances. Baking soda, for example, has pH 8. Ammonia has pH 11, and sodium hydroxide has pH 14. Pure water is neutral and has a pH of 7. So that brings us to pH in cosmetics. Why is that important? We'll first just briefly mention dental enamel. Dental enamel contains 98% calcium phosphate mineral. This is known as hydroxyapatite, and it has a critical level of pH, which is basically pH 5.5. And if you're below pH 5.5, tooth demineralization occurs, essentially if you're below that pH. Microbial growth, this is also important when we talk about pH. Microbial growth occurs best around pH 5 to 8. Isoelectric point. The isoelectric point of a protein is the pH where the sum of the positive and negative charges is 0. Okay, so essentially, proteins are composed of amino acid groups. Some of the amino acid groups have positive charges, some of them have negative charges. When you add up all of the charges, if they're all balanced and you have a 0 net charge, that's the isoelectric point. For hair, the isoelectric point is at pH 3.67. So what does that mean? That means that hair, if you're above pH 3.67, it has a negative charge. If you're below that, it has a positive charge. Also important is the acid mantle on the skin surface. pH plays an important role in this. It inhibits bacterial growth and fungal infections. In general, the surface of normal skin has a pH between 4.2 and 5.6. And the last thing that's here is classic soap bars. So we talk about hard or toilet soap. These traditionally were made of sodium salts, of fatty acids, and these had a pH ranging from 9 to 10. Whenever you use a lot of these soaps, they're considered to be, they leave films and deposits on things, and that's essentially because tap water contains calcium and magnesium ions, which can complex with the carboxylate groups of the fatty acids. These are known as sticky salts, and they're not readily soluble. So that's why they can remain not only on the surface of different things, like in the bathroom, but also on the surface of the skin. In this way, it's a form of soap buildup. We just have a few more items here to cover before we finish this chapter up. So we're on this next item, pH, acids, and bases. Just in general, we'll talk about some things that are important for us in terms of organisms and understanding like why pH is important, and then why acids and bases, what we need to know about those. And first, just to mention, one thing is acidosis. This is actually a condition or a physiological state when the pH of the blood is low. So if the pH is less than 7.4, in contrast to this, we can have a condition that's known as alkalosis, and this is when the pH of blood is too high. So anything above pH 7.4. So what we also know about acids and bases is that we have, if you remember from general chemistry, we have strong and weak acids and bases. When we talk about strong acids or strong bases, in general, this means that these are completely ionized in dilute solutions. So just, here are some examples of strong acids. We have hydrochloric acid, sulfuric acid, and nitric acid. And some strong bases are two good examples are sodium hydroxide and potassium hydroxide. So, just to keep in mind, during the semester we'll talk when we talk about hair. Hair relaxers are predominantly, or were predominantly, based on sodium hydroxide. They do use, in some cases, potassium hydroxide, I believe, nowadays. And there's one other base that's used as well, that is believed to be a little bit milder than sodium hydroxide. Sodium hydroxide is considered to be the strongest and potentially the most damaging to the scalp skin. Okay, so we didn't really mention this yet, but in general, the stronger the acid is, the easier it will give up a proton. If we look back, here we have these equations that we showed earlier. Equation 3, we left off here. We could have HA represent an acid, and then this is an equilibrium where you have A minus is the conjugate base, and then H plus, of course, is the proton that's coming off of the acid. So just in general, for this expression, you can establish an equilibrium constant. Again, this is just like what you learned in chemistry, where you had the product of the products, the concentration of those. So you have the concentration of H plus multiplied by the concentration of the conjugate base, and that's divided by the concentration of acid. And as we showed here, it's shown as an equilibrium constant on the left here, but this is also, we could call this a dissociation constant, which is shown on the right. From there, what else can we do? Well, we can go down to equation 5 and take the log of this, and we can actually derive what's known as the pKa. So you can see here pKa is equal to negative log of Ka. This is where this expression comes from. And this basically gives us a procedure, or shows us the derivation of this, but it gives pKa lets us describe functional groups in biological molecules. So if you have like amino acids, which we'll be talking about a lot, we want to know, like, okay, does this group have, what is its pKa value? So let's talk a little bit about buffers to finish up this section. Just in general, we sort of talked about conjugate acid-base pairs already. That's essentially what a buffer is. What a buffer does is it maintains a steady pH in an aqueous system. So what that actually means is that you can add base or acid to the system, and you really won't experience a very large change in the pH. So if in the absence of a buffer, the same addition of base or acid would cause a drastic increase or decrease in pH. For this class, for the purposes of what we're studying here, we're more interested in buffer systems that occur naturally in physiological systems. To illustrate this, this is showing you on this slide a titration curve of acetic acid. In a typical titration curve, you monitor pH as you add base to the system. So you can sort of monitor how much base you add, and it's recorded here as equivalence of OH minus. And what you can see here is that at low pH, there's large changes in pH as you add small amounts of base here. Okay, in the acetic acid system. However, once you reach up here close to pH 4, you can see that now as you increasingly add base across here, you don't get such large differences in pH. So this blue region here that is shown, this is known as the buffer region. You can see here it's from pH 3.76 to pH 5.76, and essentially that means that if you add base or acid in this region, you're not going to experience any very large changes in pH. What you can also see here is that at this position over here on the left, basically you have acetic acid. You can see it's in its protonated form. As you go into the buffer region, you have a mixture of the acetic acid and then the acetate anion. So you have dissociation. You have a mixture of both species present in your system. And at this point right here, where you have 0.5 equivalents of hydroxide, where you have 0.5 equivalents of hydroxide, this is where you have an equal amount of the acid in the protonated form and in the deprotonated form that's shown here. Once you leave the blue region here and you get to these highest pH areas here, this essentially you only have the anion form present. Okay. So, almost all the processes that we encounter in biological systems are pH dependent. The proteins that we'll be dealing with in the semester, they have amino groups, so they have NH2 groups or NH3 plus groups, and they also have carboxylate groups. These are ionizable, so they can change. They can have protons added to them or taken from them, so they can change charge. In a sense, these amino and carboxylate groups essentially carry out the function of weak acids and bases, and they actually act as buffers. So in doing so, proteins have built-in buffer systems automatically with them. In nucleic acids, they have phosphate groups. As you recall, we showed adenosine triphosphate, ATP. You saw that there's 3 phosphate groups there. These act as weak acids. We also know in biochemistry that enzymes are very sensitive to pH changes. Enzymes catalyze biochemical reactions, so their catalytic activity really depends on the pH of the system. And in general, the pH of the system, since it affects the ionization of side chains of amino acids and end groups, that's going to influence the ionic or electrostatic interactions with the protein, and that's going to definitely influence the 3-dimensional structure of the proteins. In the cytoplasm of the cell, we have functional groups of amino acids from the proteins, and also functional groups from the nucleotides, from nucleic acids. Okay, again, both of these act as buffers. Another one that I would like to mention here is shown in equations 6 and 7, where we have two important buffer systems. The first one is known as the phosphate system, and this occurs in the cytoplasm. It's shown here, so you have dissociation of H2PO4- to H plus and HPO42-. In the second set here, we have uh, the carbonic acid system, and again, this is a very important system, and this keeps the pH of blood very close to a pH of 7.4. You can see we have carbonic acid shown on the left here, and then we have its conjugate base, the hydrogen carbonate ion, and then we have H+. So, having this is a buffer system that is in place in the blood, and this helps maintain the pH at about 7.4. So that is all we have for chapter 1. Thank you for your kind attention. I realize this chapter was a little bit slow. Probably my presentation of it was a little too slow, but I'll speed it up in the next chapter. Or you can give me some of your feedback and let me know what you think about that, or how you would like to see things, and maybe we can make it a little bit more lively and a little bit more entertaining. Thank you, and have a good week.